This is the Group36 Senior Design Project for 2025 from University of California, San Diego, the Department of Bioengineering. This is under the EO Lab and our mentor being Sama Afe. I'm Deepta Bhardwaj and my colleagues are Margish Chodwadia and June Robbins. Our project is about micropattern multi-well plates for single rosette brain organoids at high scale. What are brain organoids in the first place? They're basically like 3D mini models of the brain itself. So as you can see, you would think like, oh, the brain is one of these big structures. Not really. It's one of these smaller pink nodes that are being formed, which is what develops into the brain in the human body. So when the zygote is formed, these smaller rosettes or structures are what develop into the brain. So why single rosette? Like I said, the rosette is the one that starts off in a single zygote and develops into the brain, not multiple of them together. Now, whenever in research we want to do something like drug testing or how a disease is going to manifest itself, we want something that's very similar to the actual human body itself than something that's not as similar. So a single rosette would be more accurate. So what would be the advantage of having a project like this that's able to develop a single rosette brain organoid, right? Compared to existing organoids that are being grown, we would be able to have increased organoid reproducibility of 70%. We would have 5 times faster screening of neurodevelopmental drugs. We'd be able to reduce modeling variability across batches by 60%, cut neural tubes defect study timelines by 50%, and improve detection of early brain malformations by 3 times. So having single rosette brain organoids would be much beneficial to the bioengineering industry than multi-rosette brain organoids. And now, is there somebody that's already doing something like this before? Yes, they're called Neuroscience. But the issue with theirs is that they aren't very consistent, and if you want to say customize something, it takes a lot of money and it takes a lot of time. So that's our project overview: having scalable, uniform and reproducible single rosette brain organoids. As you can see, here's how a single rosette brain organoid looks like, with layers of different kinds of cell forming around it. So this is how our design approach went. We saw different ways where cell patterned growth is something that's been existing in this industry for a while, and people use either MPEG or gold sputtering micropatterns. As you can see, this beautiful heart was made by a bioengineering postdoc here at UC San Diego. But this can't really help make the micropatterns that we want. And the gold sputtering one wasn't very, you can get the micropatterns, but you can't really get the cells to stick very accurately. So we thought of combining the two and make something that was similar to what is done in this research paper, having patterns and doing anti-fouling using MPEG. So these were the 2 parts of our project, and a lot of it was optimization of the PEG SH to be able to have cells stick where we want it and not stick where we don't want. So we did test, and you can see that the cells have stuck and on the glass, and cells are not stuck on the gold. So now the micropatterns itself. What's unique about us? We found a paper where that nodes or rosettes are formed in the corners. So we were like, why not get the corner at the center of it? And we simulated that using concentric circles in micropatterning. So we went through a whole design of this entire process where we go from sputtering to spin resist, direct write, and then ion milling finally, to be able to get the depth gauge to show patterns like this, where the center is lesser than the size of a cell itself to simulate. So this is how our final prototype looks like. And for the next steps, this project will be taken on by another lab, which is going to try using a new technology to be able to truly get very consistent single rosette brain organoids. So we would like to thank all of these people that have been crucial into making this project happen. And here are some memories.